We are recording. If you're watching this video, so not if you're online right now, but if you're watching this video at a later date, as always, you need to take a screenshot when I say an attendance word or attendance phrase or whatever. So for example, the first one, for those of you watching the video, not for anyone who's online right now. Of course, if you're online, you can also send this for extra credit, but your first word for attendance is going to be water. So you're going to send in the word water, and you're also going to send a screenshot. And the screenshot should show what we see here right now, which is should be like two of my squares, one of which has a circle of me, with a picture in it. And then you can see that John Raines is here too, right? That's the screenshot you need to send. There's not always a corresponding slide like one of you had mentioned previously. Just take a screenshot of when I say a word or a phrase. So yes, the first word is water. And what I see on my screen right now is what I should see from you when you take the screenshot and send me that list of attendance words with a one corresponding screenshot for each of those words. If you have any questions about it, please let me know. Other than that, we've got about 3 minutes and some change before we get started. So I guess you could skip ahead if you're watching the video 3 minutes, or you can just hang out and wait. Okay, it's 8 o'clock. Let's get started. No one's here, so that makes attendance very easy. But I just wanted to say that since we're recording. Oh, I take that back. One person is here. All right. So, nonetheless, let's get started anyway. So if you don't mind, I run a little bit behind. I don't have my attendance sheet, so you usually do it anyway, but can you email me after school, after class, let me know you were here? Thanks. Okay, let's jump back into the discussion. And then we'll get to the announcements. So let me back it up a little bit. Let's talk about some mutations. Come on. We're getting there. There we go. Mutations. This is where we left off. So we talked about everything else. And in my opinion, the most important stuff in this chapter we're already done with, which is that middle bullet point right there, other than the mutations, right? From going DNA to RNA to protein. And I say it's the most important because what I mean is you're going to have more questions about that than you will other stuff. So focus on that. But let's get started on the mutations. Before we do, let me give the first official word for attendance, or the official phrase for attendance. And it's going to be the word RNA. So RNA is the first official word for the attendance. So if you are watching this online, excuse me, not if you're online right now with us right now, but if you're watching this video at a later date, I need a screenshot of every time I say a word. And in this case, I've just said one of the words, and it's RNA. So when you send me the screenshot and along with the words, I should see, in this case, a screenshot where I have circled the particular word, which won't always be the case, but yeah, always send a screenshot from when I say one of the words.
Morning.
Good morning. For those of you who have come in after I got started, if you could do me a favor and send me an email when we're done to let me know that you were here. I was running late and I didn't have my attendance sheet.
I forgot to send you email with me.
Yeah, please do that. All right. So that being said, let's jump into this. Let's talk about these mutations. Because basically, again, we talked about, how we go from DNA to RNA to protein. And when we talked about DNA, we talked about what could go wrong with meiosis, right? When we talked about, well, that wasn't specifically DNA or this chapter, but when we talked about what can go wrong during meiosis or mitosis. So now let's talk about what could go wrong when we have this process where we go from DNA to RNA to protein. And that basically, a lot of it has to do with mutations. So you need to know what a mutation is for the exam. And the mutation is any change in a nucleotide sequence of DNA. So remember, it's those letters, right? This is a language made up of letters, A's, T's, C's, and G's. And these letters make up words. And in this particular language, every word has 3 letters. They're all 3 letters long, right? So you know about all that. So a mutation is when you change that, right? You change the words. Using the same analogy I've used before, if you think of your chromosomes, each individual chromosome is a gigantic book, right? So that means you would have 46 of them, 23 pairs of chromosomes. Each one of those books has different instructions on how to build different proteins. So a change in the nucleotide sequence, a mutation in this analogy, would be like you go in there and you change some of the letters around, right? And sometimes when you change those letters around, you're completely changing the word around, right? You're completely changing the meaning of the word. Sometimes not, but we'll cross that bridge when we get there. But yes, that is what a mutation is. You need to know what it is, or you need to know the definition. Any questions about what a mutation is? All right, and just so you know, it can be a small thing or a large thing. So for example, it can involve large regions of chromosomes, which we'll cover here in the next few slides. It can involve just a single nucleotide pair, as in the example I've already given you before for sickle cell. That wasn't an example for mutations, but sickle cell was an example of pleiotropy in the previous chapter. And of course, it could be anything in between, right? So again, the point here is mutations is when you basically change the letters in the DNA, and it can be huge, it can be tiny, or it can be anywhere in between. Any questions so far before I show you some pictures from the other textbook, the old textbook? All right, so speaking of sickle cell, this is a real example, right? Here's a gene on the left. This is, of course, not the whole gene. We zoomed in to one particular codon. Remember, codon is a word, right? So we zoomed into this gene that causes sickle cell, right? We've zoomed in to this one codon that should be CTT. which would result in the mRNA having a codon of GAA. And that codon says, bring me this, that GLU amino acid. Sorry, I forgot that my camera's not working, so I need to stand here instead of there. But anyway, that's what a normal DNA, a normal part of that gene would look like. But for sickle cell, they have one tiny little mutation, right? So instead of a T, they have an A instead in their DNA. Therefore, instead of having an A for their RNA, they have a U instead. And therefore, instead of having this GLU amino acid, they have this VAL amino acid instead. So that's just that one little change causes that whole disease, right? And we talked earlier, like I said, that we used sickle cell as an example of pleiotropy, which is when you have one allele, right? One different version of the gene. And because of that, you had all these different symptoms. So it's just a great example of mutation. Because again, you just change this one letter, and now you have this whole disease based on this one letter. And this will also be important later when we talk about natural selection. And I know you guys have already done the lab for it, but once we start talking about natural selection, it should be interesting to see how just this one gene, the one change, right, one letter in this change, we have this whole, well, you'll see about how sickle cell has become more prevalent due to natural selection. So we'll cross that when we get there. Anyway, let's make things a little bit more complicated. There are two categories of mutations in a gene, and you need to know the difference between the two of them. They are called substitutions, is the first category, and the other one is deletions or insertions, or even deletions/insertions. And the good news is they're very easy to remember the difference between because it's in the name, right? So substitution, as you can see here, well, first of all, the last one I showed you was a good example of substitution, right? So if we look at, and I'm backing up here, what we've done here is we've substituted this T for this gene with an A, right? That was a substitution. And I'll give you some more examples of substitutions, and then I'll give you some examples of deletions and insertions. But here's another one. So here we can see we're looking at some random hypothetical gene. You can see all these different codons, right? These 3-letter words, and you can see what it's supposed to be, right? We see the code up here, and we see what those amino acids code for. In this particular example, we've substituted this G for this A, and now that codes for a different word because the word is no longer GGC, the word is now AGC. And that causes us to have a completely different amino acid. And remember, if you have a different amino acid, right, you're going to change the shape of the protein. And if you change the shape of the protein, you change the function of the protein. Sometimes it's livable, right? So sickle cell is a good disease of that, or a good example of that. It's a disease. It can kill you. It's definitely unhealthy, but it's not like, you're still born, right? So substitutions can be very bad. They can be mild. It all depends on the particular letter. And later you'll see, I'm going to give you some other vocabulary words. Sometimes you can change out a word or a letter in the word and still get the same amino acid. But we'll cross that bridge when we get there. Any questions about this slide? All right, so that's a substitution. The next thing we're going to look at is a deletion, and this is a bigger deal. So again, you can see the normal, what it's supposed to be up top. Down here is what the after the mutation. So here we have a UUU, right? But in this case, what we've done is we've accidentally deleted this U, right? You can see it's out of there. So now instead of a U, it's a G. But keep in mind too, when you deleted that, this whole thing just shifted to the left. So now every single one of these letters from that U right here, from here on to the right, all of those letters are messed up. And also keep in mind, we've only zoomed into this gene. Genes are really, really long. We've zoomed into this one particular gene. So now in your mind, try to imagine how many thousands of letters there are to the right that are now completely wrong. So now we have all these different new amino acids, the wrong amino acids. We basically have an entirely new protein, depending on where the deletion is. And obviously that's generally going to be a bigger deal than if you just change one letter, which is the previous example in a substitution. Any questions about deletions? All right, insertions are pretty much just as bad. So again, we have the normal situation up top. Down here, we have the mutated version. And in this case, again, instead of having a UUU, which gives us this PHE, amino acid. Now we've squeezed this G in, right, in between this U and the U. So now it's UUG, which again causes a whole different amino acid. And again, this causes everything here from here on to shift to the right.
Where did the G come
That's a good question. It's just a mistake. And there's different ways the mistake can happen, which you can look into for independent work. But the basic answer is think about what you learned in or previously in this chapter for transcription, right? So you've got that RNA polymerase and it's reading the code and saying, all right, I need a G, I need a C, I need a C, I need a G, right? And if it gets the wrong one, it's, that's how that happens. That's actually how it happens for all of it, whether it be an insertion or deletion or substitution. Generally speaking, it's the RNA polymerase that read it wrong. But there's also some other exceptions, like the process of when you duplicate DNA, right? Maybe there was a mistake when that happened. Or maybe there was just, as you're going to learn on the previous slide coming up, not yet, but there's these things called mutagens. So if you could just damage your DNA, which would usually be more of an insertion or a deletion than an insertion. But yeah, a lot of different ways. And like I said, here in the next few slides, or after a few slides, I'm going to give you some examples, some broad examples. But for independent work, you guys can look it up and figure out specifically how does it happen, and how does your body or how do your cells deal with it? Because sometimes they are fixed, sometimes they're not. Good question. Yeah. Any other questions about the insertion? All right. Now there's some more vocabulary words. There's a lot of words for this section, right? So there's also stuff called missense mutations. And those are basically, so far, that's everything we've talked about. Missense mutations change the amino acid coding, right? So every example I've given you so far, whether it was a substitution or deletion or an insertion, it changed the sequence of amino acids, right? Because we changed the letter, which changed the word, and then that word then called for a different amino acid. So that was missense mutations. Next, the next category is something called nonsense mutations. These are mutations change the amino acid codon to a stop codon. So instead of this time, again, instead of changing the amino acid, right? It's like, oh, you're bringing me this amino acid instead of that amino acid. In this case, you're saying, we're just stopping the whole thing. And there'll probably be some sort of test question about this. Like, let's see if you guys can guess. I'm about to tell you, but in a real simple way of saying it, so don't overthink it, because that would be harder. But if you think about this really simply, what would be the result of a nonsense mutation? If you had to explain it in a very simple way. Of a nonsense mutation. Or if we had a mutation that changed what should have been an amino acid and instead now calls for a stop codon. So this requires you to remember what a stop codon is. How's that? Sort of, yeah. It wouldn't form past that point. So basically, and so you're on the right, that's basically what I'm getting at. And that's what the test question is going to be, something like that. And the answer is it would result in a shorter polypeptide. Because remember, these codes, right, we're bringing in amino acids to build a polypeptide. So then if you accidentally get a code that says, all right, stop doing that, then the result is going to be a shorter strand of polypeptides. So yeah, that's what you're going to need to remember. And in the exam, it probably won't, obviously, it's probably not going to remind you that a nonsense mutation is a change in the amino acid codon to a stop codon, right? It's probably not going to tell you that. It'll probably just say, what is the result of a nonsense mutation? So that's going to require you to know what a nonsense mutation is. So any questions about that second bullet point? Okay, as we mentioned earlier, a deletion or insertion, which is also, by the way, I haven't mentioned this yet, is also known as a frame shift mutation. And that'll make a little bit more sense on the next slide. I'll give you a visualization. But those often have disastrous effects, like I mentioned earlier, right? So a substitution, yeah, you're changing one letter, which might change one word, which will change one word, which might change one amino acid. But when you delete something or insert it, remember, you screw up everything all the way downstream, right? Because everything gets shifted, hence the name frame shift. So that's something else you need to know for the exam. If you had to choose which one is worse, deletion/insertion or mutation, substitution, then the deletion or insertion would be worse. Yeah, and as it points out here, it usually produces a non-functioning polypeptide. So when you have that mutation, it's usually just like, well, this is not working anymore. Because like I mentioned earlier, it's basically a brand new protein, depending on when the frame shift mutation happens. So for example, and I'm not going to get this deep on the exam, imagine if that frame shift mutation was at the very end of a gene. Like, yeah, so what? You might mess up one amino acid, maybe two, depending on where it is. So, but yeah, it depends on where it is. All right, so the next word for attendance, and you should take a screenshot if you're watching the video. So the screenshot should show this slide, and I should see the underlined frame shift and the circled bullet point at the bottom left. But anyway, the next word for attendance is to answer me this. What kind of mutation causes a change in the amino acid codon to a stop codon? So there's two different types of mutations we mentioned here. Well, I guess there's three, but of the types of mutations that are on the screen, which one results in a premature stop codon is another way of saying it. It's the only one on the screen that mentions anything about stop codons. So there you go. That's your next word for attendance. All right. Before I take any questions, I'm going to go to the next slide, which is a picture of this frame shift mutation. This is not from your textbook. It's from some other textbook, but it's a great picture. So you can see here, like, what the words are supposed to be, right? It's supposed to be CAT, TCA, CAC, GTA, right? All these words that code for these particular amino acids. And then what happened is, right, these things all shifted one to the right, as you can see down here, right? So instead of being CAT, it is now ATT, right? You can see everything. And now this whole thing has shifted, right? So now we have this whole new strand of amino acids, which weren't originally supposed to be there. And again, this is why they call it a frame shift mutation. You can see how the whole frame has shifted. Speaking of which, in theory, if you think about it, in theory, if you had some sort of frame shift mutation that shifted by 3, that would be a lot less disastrous. I don't know if that would ever happen. I don't remember. But think about it. Like if you just deleted 3, if you had 3 deletions, you would miss that amino acid, but then the rest of them would stay the same. Or if you had 3 insertions, right next to each other, then yeah, you would have a new amino acid, but everything else would remain the same. So again, I guess that would be one of a very rare circumstance. But any questions about frame shift mutations? Here's a nice little table that I really don't think is in your textbook. So I recommend looking at this PowerPoint to study it. But I don't know, again, everyone's brain works different. For me, I like to see things summarized where I can compare and contrast them. So I love tables. But here we go, right? The 2 different types of mutations. You've got substitutions is one category, or insertions and deletions. Under substitutions, you have these different effects. A silent mutation, and I haven't talked to you about this one yet. So a silent mutation would result in no change to the amino acids. Now, I wish I had that little chart. I don't know if you remember that chart I showed you from the other day that shows you how to translate RNA, right? And I don't remember the exact words or the exact order of the word letters, but it could have been like, I don't know, A, U, C, G, right? And then it had another little wheel and then another little wheel, and you would go out to determine what the amino acid is. The point here is, for a silent mutation, this is one of those situations where you change a letter in the code, but remember, there's 64 different words, but there's only 20 amino acids. So for each one amino acid, there's more than one word, more than one codon that says, bring me this amino acid. So what happens in a silent mutation is you've changed the letter in the word. So you've changed the word, but the word is still calling for the same amino acid. That would be a silent mutation. You change the letter, but it still codes for the same amino acid. Therefore, as far as the protein is concerned, it's a silent mutation. It didn't make a difference. Missense, we already talked about. So just again, as a quick reminder, that's when you have one amino acid for the other. Again, nonsense, which we already talked about, says that's when you have this premature stop codon and it stops the whole thing in its process. And of course, insertions or deletions are also have the effect of frame shifts. So again, this is everything we just talked about in a nice little table, but are there any questions? All right, so to answer the question previously asked, we'll just scratch the surface of it. How do these things happen? First of all, let's talk about mutagens. Mutations occur in a number of ways. as I mentioned earlier, first of all, there's errors in DNA replication. So again, think about what happens during S phase of interphase, where that DNA polymerase attaches to the chromosomes, right? And it duplicates the chromosomes. It's opening it up. It's reading the code. It's building the new strand based on the code. But sometimes that messes up. And that's one of the ways we have these mutations. Of course, there's recombination. So if you remember when we talked about crossing over, for example, when we talked about prophase 1 of meiosis, when those homologous chromosomes are really close together, sometimes they swap chunks. That's crossing over, right? Sometimes when they do this swapping over, as we mentioned in that chapter, so I'm not going to get too deep into it now, but it happens in wrong ways, right? Sometimes things get inverted. Sometimes huge chunks are missing. Sometimes you have huge extra chunks, right? All of this can happen during DNA replication or recombination, which again would be like crossing over, when crossing over goes bad. The second one here, second main bullet point, is physical and chemical agents called mutagens. These are the most common physical mutagen is high energy radiation, such as X-rays and ultraviolet light, right? That's why when you have X-rays, you're supposed to be wearing protective lead bib or whatever it is. It's supposed to protect you from it. UV light, right? That's why they say you're supposed to, not avoid the sun completely because you do need it, but protect yourself from it, right? Wear clothing, sunblock, stay in the shade, stuff like that. Chemical mutagens. So that's like, think about tobacco, or not tobacco itself, but think about just cigarette smoke in general is a great example. There's so many different carcinogens, and they are mutagens, but they call them carcinogens because not only do they mutate things, but those mutations can lead to cancer. So obviously you can see we've just scratched the surface. This is a wealth of opportunity here for independent work. There's a lot of stuff you can write about. So other than X-rays and ultraviolet rays, what are some other physical mutagens? Or if you want to, how exactly do X-rays cause mutations? How exactly do ultraviolet light cause mutations? Chemical mutagens. Again, I didn't even give you any specific examples. You can look that up for independent work. And we have a lot of them in this building, actually. But don't worry, we keep them safe. So anyway, yeah, you can look up a lot of that for independent work. The point here is that you know what a mutagen is, right? It's something that causes these mutations. It can often be harmful, but sometimes it can be beneficial in nature, and definitely in laboratories. And you can look that up in Nature or for independent work if you want. How is that beneficial in labs? And long story short, it's because sometimes we want to cause mutations because we want to see the result of it. But that was a very basic description. So if you want for independent work, you can go find some specific examples where in a study they caused a mutation to a gene and then this was the result. Right? That actually used to be a lot more prevalent. Well, actually, I don't know. I assume it's less prevalent now. But anyway, before we had GMOs, before we could specifically edit genes, that's how they used to do plants, right? They would just like mutate a bunch of plants and see which ones had something beneficial. Like, let's start growing that now instead. But now, we're able to specifically pick a gene and fix it. But anyway, mutations are a source of rich diversity of genes in the living world. And it's because the these mutations that we have this diversity that makes evolution by natural selection possible. So once we get into natural selection, just know it's these mutations that make it all possible. So since you've already done the lab for this week, it's that mutation that one moth had that randomly turned some of them black. And then therefore, the timing was perfect, right? They happened to turn black. Meanwhile, the surroundings happened to turn black because of all the pollution. Therefore, all of a sudden, these black things had an advantage. So yeah, that'll definitely be a test question later too. Once we talk about natural selection, I'll bring that back up. But some question about like what's the source of all the diversity that causes natural selection, and it's mutations. Without mutations, we wouldn't have natural selection. There would be nothing different, right? We're all different because of mutations. Anyway, any questions about this slide? Okay, let's do announcements and reminders. Largely, I'm kind of preaching to the choir because if you're here right now or if you're joining us online or if you're watching the video, you're doing what you're supposed to be doing. But let's think about this. Some of you really need to turn things around. And I'm trying to put myself in your shoes. And I can see how some of this can be very overwhelming. Like, man, what am I supposed to do to this lab? What am I supposed to do with the attendance words? So if you just go back to basics, we're going to be okay. right? Come to class, come to lab, do your assignments on time, and read the course announcements. Because all these little extra things that I'm asking you to do, those are for people who aren't doing what they're supposed to do, right? Oh, you can't physically be here for class. Well, log in online, make sure you send these attendance words. Oh, you can't come in and log on online. Well, you can watch the video. Make sure you send the attendance words. Make sure you send the screenshots. Oh, you didn't do your pre-lab in time? Okay, well, do this to make up for those points. Oh, you couldn't make it to lab? Okay, well, do this to make up for it, right? Oh, you didn't do it on time? Well, you can do this to make up for your lost points, right? All these extra little things that I'm asking you to do, these are only for people who aren't doing what they're supposed to do. So just do what you're supposed to do. You won't have all these problems. And again, I do realize it's overwhelming for those of you who have not been doing what you're supposed to do. Therefore, you've got to jump through these extra hoops. So even though it's not written on the screen, I'll remind you, I'm here to help you. Come meet me online, Big. I don't even remember what I'm supposed to do for this lab because you've asked me to do so many extra things because I didn't come to lab like I was supposed to. Meet me online. I'll walk you through every little step. I'm here to help you. This one's a reminder. I've been saying it for a while, and you guys here. Don't forget also, speaking of which, to send me an email today for those of you who are here because I wasn't able to get my spreadsheet pulled up in time. But anyway, everybody who comes to lab physically now, lecture now gets 4 times the extra credit. So 4 points instead of 1. So please take advantage of that. Here's a new one. This is an announcement, not a reminder. I have office hours. My office hours are 100% online today. So when we're done with class, I have to go home. My kids are on spring break. My wife has a meeting that she has to attend, so I have to be there. So I will be not in my office today, but I will be online from home. What's that? Okay. How do you set that up?
For office hours.
Oh, yeah. Send me an email and I'll send you the link. Yeah, it's a link I've shared before, but it's a long time ago, so it's lost in a bunch of other stuff. Exam 2 is still open for retake. Again, take it as many times as you want. There's nothing to lose. Just make sure you let me know when you're done. It closes when exam 3 closes, which I don't remember exactly when that is, but I would assume it's coming up.
The 7th, I think.
Oh,
When it closes, my fault.
yeah. I'll check. If it's anytime soon, I'll make sure to send out an announcement. Otherwise, I'll just remind you guys on Monday. But yeah, make sure you do that. Again, independent work. Some of you haven't done anything. I'm begging you, at least write 1 paper so you can see how I grade. Don't wait until the due date and write a bunch of stuff thinking you're going to get 100, and then it turns out you get a 0 because you didn't do what I asked you to do, and therefore you got a 0, and now it's too late for you to fix it. Please write at least 1 paper. And remember, could be 1 sentence, could be 100 pages. It doesn't matter. Write a paper. Make sure you include your sources and send it in so you can see at least how I grade. And also to make your life a lot easier when you have to get 100 points by the last day of class. Might as well start now. And then finally, a reminder, lecture and office hours will be 100% online the Friday before Easter and the Monday after Easter. So any questions about announcements and reminders? To reiterate the answer to the 1 question. If you don't know how to meet me online, send me an email and I'll send you the link. All right. Let's jump into this. Let's talk about virus and other non-cellular infectious agents. We should be able to get through this pretty quickly. Actually, let me take that back. Let's see. Yeah, that'll be good. Let's move forward. Let's talk about viruses and other non-cellular infectious agents. Viruses, that's what we'll talk about first. Viruses share characteristics of living organisms in that they have genetic material in the form of nucleic acids. It's packaged in highly organized structure, right? We have that in common. But we generally don't consider viruses alive because, first of all, they're not cellular. And if you remember way back to chapter 1, when we talked about the characteristics of life, that was one of them. Life is cellular. And another one, it was reproduction, right? Life reproduces. Viruses do not reproduce, at least not on their own. They have to hijack your cells or some other living organism cells to reproduce. So you need to know that about viruses. They're not alive for those 2 reasons. They're not cellular and they don't reproduce. They are similar to us in that they have the same, they have nucleic acids, right? They have genetic material in the form of nucleic acids that is packaged in a highly organized structure. So know the similarities, which is the first bullet point, and the differences, which is the second bullet point. Notice too, in that first bullet point, I said nucleic acid. I didn't say DNA or RNA. And you'll see why in the second. Well, I'll just go ahead and tell you, but then officially tell you later. It's because viruses, some of them are DNA, some of them are RNA. It depends on the kind of virus. So again, viruses are infectious particles consisting of genes in a box. So it's basically the same thing I said on the previous slide. Now I'm just saying it in a different way. So genes in a box, right? Like I said, nucleic acids in a highly organized form. It's almost always a nucleic acid wrapped in a protein coat. So whether it be RNA or DNA, it's one of the two, and it's wrapped in a protein coat. And in some cases, those nucleic acids that are wrapped in a protein coat are also have a membrane envelope, which makes it even closer to being a cell, right? It's still not a cell, but it's close because it has a membrane, just like a cell does. But more importantly, I would say, as far as the exam is concerned, if anything, these 2 bullet points are probably more important than that third one. Granted, I'm probably also not going to get that detailed on the exam either about asking you about genes in a box and nucleic acids wrapped in a protein coat. You do need to know this. And again, I've already said it, so here we are saying it again. Viruses cannot reproduce on their own. For that reason, they have to infect a living cell, and they direct its molecular machinery to make more viruses. So all that stuff you just learned about, from DNA to RNA to protein, right? Transcription, translation. Viruses hijack that. They don't have that ability. So they take their genetic information and they use our cellular machinery to turn that genetic information into proteins and other stuff. And we'll talk about that later in this chapter. Let me think. All right. So the next word for attendance is either yes or no. And the question is, can viruses reproduce on their own? And of course, if you're watching the video, you're going to have a screenshot. I should see that in this case, this slide that has me writing on it, with a little box and a little check mark. All right, let's move forward. Here's kind of what I'm talking about. Here's a picture of what we're talking about. In this particular example, we have DNA instead of RNA. You can see you've got this DNA in there, and it's inside this little box of protein coat, right? That's a good example of a virus. Now let's talk about different types of viruses. So viruses are not equal. They attack different things. So not every virus out there attacks humans. Not every virus out there attacks animals in general. So before we even talk about this one, does anyone want to guess what bacteriophages attack? If you look at the screen, if you had to guess, these viruses attack what? No, what kind of cell do you think these things attach? Which is a good question. I'm glad you said that. Even though I'm not going to ask you on the exam, generally speaking, all viruses basically attach the cell membrane. They all have different ways. Usually we've got some extra stuff on our cell membranes that they attack to get to the cell membrane. But what I'm saying is not all viruses infect humans. Not all viruses even infect animals. So what kind of organism do you think bacteriophages infect? There you go, bacteria. There you go. Yes. These are viruses that attack bacteria, hence the name bacteriophages. Phage or phage, you can say either one. Phage or phage, that means eat, right? So these things eat bacteria, basically. And generally, sometimes people call them phages for short. But yeah, and they consist of DNA enclosed in a structure of proteins. And this is an example of one right here. This is a good drawing. So, and if you click it, there's a little video you can watch that gives you some more information about bacteriophages. Even though I'm not going to get into it on the exam, you need to know what they attack, and it's in the name. But for the most part, you don't have to know much about it. There is one more thing you're going to need to know, and I'll let you know when we get there. But yeah, that's what one of them looks like. Here's a picture from a scanning electron microscope. This shows 3 of them attacking a cell. So you can see the 3 of them up top. You can see at the bottom that kind of gold looking color, that's the cell. And you can see this green stuff coming into the cell, right? It's almost like a hypodermic needle. So they attach to the cell in this particular case, and they don't all look like this, but in this particular case, they attach and they just kind of inject their genetic material into the cell. Here's a better or a different drawing. Here we can see, and you can see too, this one looks a little bit different than the last ones, but very similar. Again, almost like a hypodermic needle, right? It attaches and injects its genetic material into the cell, which is colored in red. You can see the green DNA, or yeah, it is DNA. The green DNA in this case is the original bacteria DNA. So you can see that this red DNA incorporates itself into the bacteria DNA. And then a bunch of other stuff going on over here that we're about to talk about. But eventually, basically, long story short, before I give you all the details coming up, what happens is, again, it injects its genetic material, which is instructions. Remember, genes are instructions on how to build stuff, particularly proteins. So it injects its genetic material, the cellular machinery inside that bacteria. In this particular case, it happens to be an E. coli. It reads the instructions. It builds the stuff. It builds so many proteins that eventually the whole cell just busts open because there's so many viruses, inside of the cell. It busts open and then they go off to infect other cells. But again, that's just a picture of it. What we're about to do is talk about it in more detail. And we'll have some letters here, some words on the screen. So here we go. Once a bacteria is infected, most of the time it enters a reproductive cycle called the lytic cycle. So like I said, I'm not going to ask you many questions about this on the exam, but this is one of the questions I'm going to ask you. You will need to know the difference between a lytic cycle and a lysogenic cycle. So I'm about to explain both of them. Right? So again, usually it enters the lytic cycle, and I'm going to explain more about the lytic cycle here in a second. But sometimes it reproduces by an alternative cycle, which is called the lysogenic cycle. So again, let me explain the lysogenic cycle, then we'll go back and talk about the lytic cycle. In the lysogenic cycle, the viral DNA the viral DNA is replicated without a phage production or the death of the cell. Another way of saying that is it injects its genetic material, like I've already said, but in this case, instead of the cellular machinery reading that genetic material and building a bunch of viruses, instead this time it's just like, all right, well, let's not worry about it. And the DNA incorporates itself into the bacterial DNA, and it just stays there. And then every time that bacteria goes through mitosis and reproduces itself, remember, it makes a perfect copy of its DNA, right? So every time it does that, it makes a perfect copy of its DNA, including the viral DNA that is now incorporated into it, right? So even though I'm not going to ask you this on the exam, this lysogenic cycle, we use that a lot in this building. If we want to make a copy of a gene, we often use E. coli. We inject it, the particular gene that we want, we use a virus to insert it into an E. coli, and then it reproduces like bacteria do, right? And every time it reproduces, it's also reproducing a copy of that gene. And then by the end of it, we have thousands, maybe millions of copies of that gene. But anyway, that's how we take advantage of it. But yes, that's also what happens in nature. When it's the lysogenic cycle, again, its genetic material is incorporated into the bacteria DNA, but it doesn't make the viruses. It just reproduces and reproduces and reproduces, just makes the copy. Technically, there's a word for this. When this viral DNA is inserted into the bacterial chromosome, it is then referred to as a prophage. I might or might not ask you that, but yeah. No, I take that back. I definitely won't ask you I shouldn't say definitely. 99% chance I'm not going to ask you what a prophage is, but I do think there's a question that uses that term. So make sure you know what that is. So again, lysogenic cycle. Viral DNA gets inserted in the bacteria, but it's incorporated into the bacteria's DNA. And at that point, we call it a prophage, the bacterial DNA itself. Anyway, this makes more sense when I show you some pictures, so I won't take questions yet. Also, if you download the PowerPoint, you can watch this little animation of it happening. Same here, another little animation of it happening. But here we go. First of all, let's talk about the lytic cycle. Like I said, I was going to explain that to you. I kind of skipped over it. Lytic cycle is pretty simple. It's exactly what you might imagine. You can see there at the top right that the virus inserts its DNA into the bacteria, right? And then you can see here in this picture right here, that blue big blob is the bacterial DNA. That little green blob is the viral DNA. You can see it has not incorporated itself into the bacterial DNA. They're two separate things. So anyway, because of that, the cellular machinery inside the bacteria reads the virus code and builds whatever it needs to make those new proteins, or those new viruses, right? And those new viruses are assembled, and eventually there's so many of them
as you can see in this picture, right? There's more and more of them. And finally, there's so many that the whole cell explodes. And then those newly built viruses go on to infect other cells, and the cycle continues. So any questions about the lytic cycle? Okay. The lysogenic cycle, which is the one I've thoroughly described in writing, but here it is again. Here we have the virus at the top left. It's, again, just like we saw previously, so this is similar. It injects its DNA. You can see here that we have separate green viral DNA and blue bacterial DNA. But then right here in this part, number 5, we can see that this viral DNA is now incorporating itself into the bacterial DNA. So now, as we can see in part 6, every time that bacteria duplicates itself, it's also copying not just its DNA, but that viral DNA that has incorporated itself into the chromosome. And that's it, right? So again, it goes through this many times. So now you have thousands or maybe millions, maybe even billions of cells that now have this viral DNA lying dormant. So then what happens is you bring the two together. The lysogenic cycle, right, again, eventually just keep doing that. But then sometimes, and you can look this up for independent work to see what causes it, something triggers it, and then it leaves the lysogenic cycle and then goes into the lytic cycle, right? So we have all these cells, you can see here at the top right, that just have that viral DNA in them, and they're not doing anything with it. But eventually they do, right? And they jump into that lytic cycle where they do express the code written in those genes so they can build the proteins, and then the cell explodes. Anyway, again, for the exam, you need to know the difference between lysogenic cycle and lytic cycle. The way I always remembered it as a young student was the word lysogenic is a lot longer than the word lytic, and the process is a lot longer too. So if you think about, one of them is like basically instant death. That's the short lytic cycle. One of them is a lot longer because it's reproduce and reproduce and reproduce, and that's the lysogenic cycle. So if you can remember, the longer word describes the longer cycle. Hopefully that'll help you. Any questions about this slide? Also, I don't think this picture's from your textbook. I think the pictures from your textbook are not as good as these. So if that picture helps you study, then you should use the PowerPoint. And also, speaking of that picture, I should point out, you don't need to memorize this, right? There's not going to be any questions where you need to know, all right, the lytic cycle starts with stage 1, which is then goes to stage 2, and then stage 3. I'm not going to ask you all that stuff. Just know the difference between lytic cycle and lysogenic cycle. All right, plant viruses. I'm not even going to ask you this time. Surely you've seen the pattern. Guess what plant viruses attack? They attack plants. It's interesting. Plant viruses usually aren't deadly to plants. They usually just stunt the plant growth or diminish crop yields. And most of them happen to be RNA. So you might have, if anything, I might ask you those questions about a plant virus. Many of them, like the tobacco mosaic virus, are rod-shaped that have a spiral arrangement of proteins surrounding the nucleic acid. You can see that picture at the top right. That's what a tobacco mosaic virus looks like. And just so you know, it affects tobacco and related plants. I'm not going to ask you any of this on the exam, so if you want to, you can look that up for independent work. What kind of related plants? It causes discolored spots on leaves, and I think the only reason your textbook even brings it up is because it was the first virus that was ever discovered. I'm going to put it next to that just as a visual reminder that I'm not going to ask you any questions about the tobacco mosaic virus. But yeah, that's some interesting things about them. And again, stuff you can look up for independent work. Any questions about this slide? All right, the next slide is a picture from the old textbook. You can see the tobacco leaf on the right has discoloration, but then again, if you don't know what a normal tobacco leaf looks like, you can't really identify the fact that it's discolored. Anyway, and then to the right of that picture is a long rod, and that's the whole virus. You can see the little box drawn on it right here. So that's what this picture is all the way to the right. We've zoomed into that box. You can see this RNA is spiraled around into this rod, and you can see it's coated in protein.
So is that like, they're not used to tobacco leaf no more, or is it just discoloration?
I don't think they can, but that's a good question. That's a really good question. I don't know. A good topic for independent work. Not just with tobacco either, but what about those related plants that I didn't even get into, right? Can we use them? Yeah, great question. Moving forward. Now that I'm going to get this deep into this on the exam, but the book did. So here we go. I'm going to get through this really quickly. Plant viruses, they need to get past the plant epidermis, right? So it's not like they have plants don't necessarily have a respiratory system like we do, right? They don't have a digestive system like we do. So we have all these different ways for viruses to get inside of us. They don't. So a damaged plant is more likely to be infected, right? Because when if a farm implement, you're cutting something, because you're harvesting this part of the plant, that's one way it could spread. Insects can carry a virus, and you can look that up for independent work, like what particular insects carry which particular viruses, if you want to look that up. But again, I'm not going to ask you on the exam. Same thing for this. I'm not going to ask you this, but just so you know, there is no cure for most viral plant diseases, for most. So again, for independent work, you can look that up. Which viral plant diseases are there cures for? But anyway, since there are not any cures, agricultural scientists generally focus on prevention and also breeding crops that basically can resist it. So as far as the exam is concerned, that last slide is very unimportant. Any questions about that? All right. So let's talk about animal viruses. Obviously, this is the one that's more important to us because we are not bacteria, we are not plants, we are animals. So animal viruses are the causes of many diseases. So far, when you look at the entirety of human history, none of them have a greater health threat than the flu, which blows people's mind today because now we have ways to prevent the flu and ways to fix it when you get the flu. But yeah, as far as human history is concerned, that's the most deadly virus ever, like of all time. It's been around for a while too. Anyway, like many other animal viruses, the flu virus has an outer envelope of phospholipid membrane that has spikes of protein, and that membrane helps it come into our cells. If anything, what's important for this slide on the exam is I'm not going to ask you any questions about flu necessarily, but as far as we're concerned, animal viruses have an outer envelope of a phospholipid membrane that has spikes of protein. So you might need to know that. As far as we're concerned, all animal viruses have that.
Now they get in.
Yeah, exactly. Because they kind of like merge with our phospholipid membrane. So rather than inject it in, like in the example I showed you with the bacteriophage, it kind of merges in. But if I ask anything, it'll just be one question. Another thing I'm not going to ask you is to differentiate between these things I'm about to tell you. I'm not going to ask you which is which, but just so you know, some RNA viruses include flu, common cold, measles, mumps, AIDS, polio. This was made before COVID. But obviously, if COVID is related, COVID-19, right? It's related to the common cold itself. So it's an RNA virus. Some DNA viruses, some examples include hepatitis, chickenpox, herpes. I'm not going to ask you to, again, for the exam, you don't need to know which is which. But that's also a short list of viruses. For independent work, look up some other viruses. And just tell me. That'd be nice and easy, right? You don't even have to get into the virus that much if you don't want to. You just tell me, is it a DNA virus? Is it an RNA virus? Here's some pictures of what we're talking about. This is a generalized, they all look a little bit different, but this is like a general picture of an animal virus. You can see it's very similar to that plant virus, right? Here's that RNA spiraled around and covered in a protein coat, just like the plant virus. But now it's also surrounded in this membrane, right? Like almost like a cellular membrane, so it can help attack our cells. We won't have time. We got 2 minutes. We won't have time to talk about this. So there's three numbers on this slide. And the last word for attendance, it's multiple choice here. It's 1, 2, or 3. So you tell me which one is uncoding, right? So there's 3 numbers, and each of those 3 numbers explains one of the 3 things happening. Which one of the 3 is uncoding? So that's your last word for attendance. Other than, as always, don't forget to ask me a question about the materials presented. And of course, this one time, even you guys who are here, please at least send me an email that says you were here. Forget the words if you want, but just make sure you remind me that you came in. And that's it. You guys have a good weekend. I'm driving home. I'll be online if you need me.